All right, let's continue talking about the circulatory responses to exercise. In the last video I just discussed, the overarching question that I want you guys to think about as we go through this. I want you to think about why VO2 max and exercise capacity decrease so much with heart failure. So, in terms of outline, here's what we're going to talk about. First, just the major functions of the circulatory system, and then we'll talk about the organization of the circulatory system and the cardiac cycle. Most of these things should be pretty much reviewed from previous classes, but we'll go over them anyway. One of the key things that you're going to learn in this unit is this law right here. This is called Poiseuille's law. It helps us calculate flow through a tube, and we'll talk about the different parts of it throughout this lecture. I won't go into detail about it right now, but by the end of this lecture, and definitely by the time you're going to take your next exam, make sure you can explain what's going on in this equation. This is one of the most important equations for this semester. So understand it, know what it means, memorize it. And that goes for the rest of the equations. This test coming up is very equation heavy, and you want to make sure that you understand these equations, memorize them, and know how to manipulate them. All right, so what are the major functions of the cardiovascular system? A lot of it comes down to transport. Well, pretty much most of it, if not all of it. One function is to transport oxygen and nutrients to tissues in need, like skeletal muscle or the heart or the brain. It also transports and removes CO2 and other waste products away from tissues. And finally, one we aren't going to talk about too much in this chapter about the cardiovascular system. We'll talk about it more in chapter 12 when we talk about heat regulation. But the cardiovascular system also transports heat. And dilating blood vessels in your skin or constricting them is a way that your body either lets go of heat or keeps heat in, and it's very important. So cardiovascular system transports stuff. Now, how does it transport? What determines the flow? Remember, I just talked about Poiseuille's law. If you speak French, you probably are saying, wow, this guy doesn't know how to say French words, and that's true, I don't. Unless you ask some of my family members when I'm doing housework at home, like working on the house, sometimes I learn French. And I'm sorry about that. We all have our weaknesses. But anyway, that's a tangent. It's amazing. I can get on tangents even in videos when no one's talking to me. All right, so Poiseuille's law. Flow. The flow through a tube. You might have learned this in physics. Flow through a tube is determined by the pressure from one side of the tube to the other. So the triangle is a delta symbol. That means change in pressure. Delta P, change in pressure. So if we had 120 over here and 80 over here, the change in pressure would be 40, and flow always goes from high pressure to low pressure. What would happen if we had 120 to 120? Now there's no gradient, no flow, right? You always need a change in gradient. That would be 120 minus 120 is 0. We put a big 0 up here. 0 times anything is 0, so we'd have 0 flow, right? So you always need a pressure gradient. That's one major factor. Now, another thing going on, you have pi, which is just going to act as a constant. But then the major factor in dictating flow is radius, the radius of the tube. And notice that it's to the 4th power. So what that means is that a very small change in radius will give you a huge change in blood flow. Let's say we doubled the change in pressure or we doubled the radius, they would have drastically different effects. Doubling the pressure would double flow. Doubling radius would do something like increase it 16 times, right? So small changes in radius have a big change in flow, and that's one of the things that your body's really going to manipulate is that radius. Now these other factors, this is an 8, just another constant, nothing too important here. The N is viscosity of the fluid. So if you have very viscous blood, it's going to flow more slowly. If you have very thin blood without a lot of red blood cells in there, it'll flow more quickly. During exercise, we don't really see this viscosity change all that much. If you get dehydrated, it might change a little bit, but that's not going to be a major source of change. The L is the length. The length of the tube that the blood has to travel through. One thing I want to clarify before I go on is the radius. If we're talking about blood flow through a whole muscle, not just one artery, right? Your arteries will split. You start with one big artery, just like in the lungs, it bifurcates, that bifurcates, that bifurcates, each one of those bifurcate, and you just end up with millions and millions of arterioles and capillaries and all of that. When we're talking about the radius for a muscle or for the whole body, this is going to be the total radius of all of the arteries at that level in the muscle. So if you have a lot of arteries, a lot of capillaries in your quadricep, that's going to help you have greater blood flow. If you only have a few arteries or capillaries in your quad, you're not going to get a lot of blood flow in the quad. If you're just wanting to look at flow through the aorta, then the aorta would just be the radius of the single aorta, right? Okay, so again, the delta P is the change in pressure from one side to the other. R to the fourth power, or radius of the tube, and that can be the radius. If you're looking at an individual tube, that can be just one little radius of an individual tube. But if you want to look at like a whole muscle, then you look at the sum of all of the radii. Is that plural? Radii? That sounds right. I'm sure some people are rolling their heads right now. All right, and then viscosity and length, not going to change all that much. Most of the time, very constant. But you could see scenarios in pathological situations where those might become important. Now, let's talk about the organization of the circulatory system. All right, first off, why did I talk about all this? The circulatory system is all about flow and transport, right? And these are the variables, primarily these two, that your circulatory system is going to manipulate to deliver heat, to deliver oxygen, to deliver CO2 where it needs to go. It will manipulate pressure changes and radius changes, and that's how it dictates what gets transported where. This Poiseuille's law is super important for that. Now, how is the circulatory system organized? You guys should know this. I'm not going to go into too much depth in this. What I do want you to do is, in a second, pause and review this. Make sure you know the pathway of blood. What's going on from the right atrium to the left atrium, where that goes. It goes to the lungs. And then how that comes back here. Whoa, not there. How the blood from the lungs comes back into the... Where's that coming in? I can't figure out this diagram. It comes in here. I should have just left this to you. Into the atria, then comes out through the left ventricle out to the body. Make sure you know when blood is oxygenated and when it's deoxygenated. I don't expect you to know the name of all the valves and all of that or papillary muscle versus that. That's more detail than we really need to understand. Just know the pathway, where blood goes, and what the job of each of the chambers is. So make sure you remember that. So we talked about the heart. Hopefully you just paused and reviewed it. Unlikely that you actually did. So I'm going to guilt trip you until you actually do it. So that's the heart. But now if we look, the circulatory system is everything beyond the heart. And it involves arteries and veins. Now, the arteries are stiffer. And let's just ignore the pulmonary circulation for right now. We talked about that last week. So this is called systemic circulation, or the systemic circuit, which is the vast majority of your arteries. The systemic arteries are going to be carrying oxygenated blood, and they're going to deliver it to tissues in need of oxygen, glucose, and all of that. The arteries are stiff and they don't collapse. Part of that is because they have really high pressure. The blood coming out of the heart, the pressure is, the standard is 120 over 80, right? Systolic pressure 120, so during the contraction 120, after the contraction 80. So high pressure. The veins, on the other hand, they're floppy. They don't need to be stiff because they have low pressure. The blood makes it all the way down to these little capillaries, and in the capillary, you have so many capillaries that the blood has plenty of room to go, so it's very low pressure in the capillaries. Once it's captured back into the veins and venules, then you have the blood in one spot again, but the pressure is still pretty low. The veins are floppy. They're often called capacitance vessels, meaning that they can expand or get collapsed depending on if you need blood to be stored there. They have the capacity to expand and store more blood. So the veins are floppy, and they take deoxygenated blood back to the heart. In the pulmonary circuit, everything's reversed. The arteries carry deoxygenated blood, and the veins carry oxygenated blood. But for the rest of this unit, we're going to focus mostly on the systemic circuit. Now, what are some key differences between cardiac muscle and skeletal muscle? Fortunately, all that stuff that you learned at the beginning of the semester about skeletal muscle, most of it applies still. We still have actin and myosin doing the contraction, and in the heart, there are a couple of differences going on that we'll talk about, but it's the same idea. We have the sarcomeres and all of that. So let's talk about key differences between heart muscle and skeletal muscle. So as we said, we have the contractile proteins. The shape of the muscle fibers is different. In your skeletal muscle, like in your sartorius muscle or something like that, you can have muscle fibers that are several centimeters long. That's a single cell that's several centimeters. And because of that, because they're so long, they're multinucleated. They have multiple nuclei. Cardiac muscle isn't that long. They're pretty small, short, stubby cells. And actually, instead of being one long tube, they can actually branch. And that's what you can see here, kind of. So here's one cardiac cell. It branches into two. You can see that, kind of. So it branches. And they're smaller. Because they're smaller, they can get away with just having one nucleus. They have Z discs. They're striated, actin and myosin with the sarcomeres. A really cool difference and a unique adaptation with the heart is that they have cellular junctions called intercalated discs. Now, the difference or the function of this is that action potentials can be transferred from one cardiac cell to the next, to its neighbor. In skeletal muscle, when you generate an action potential on one skeletal muscle fiber, the neighbor doesn't know it. It's completely asleep. So you could have one skeletal muscle fiber contracting, and you would not have the other one contract unless the nerve innervating it told it to contract. It's different in the heart. When one action potential generates on cardiac muscle 1, so let's say we have a couple of muscle cells here connected. This would be our gap junction. If we generate an action potential, I'm going to try to do lightning here. Generate an action potential on 1, that voltage goes through the gap junction and causes the action potential to continue on to the next one. So we would have action potentials travel all through the heart. One action potential, usually at the SA node, will then ultimately result in action potentials in the rest of the cardiac muscle cells, causing the heart to contract in a very coordinated fashion. What else do we got going on that are differences here? In terms of energy production, your heart is super aerobic. If it was fiber typed like skeletal muscle, remember you have your type 2 fibers, which aren't very aerobic. You have your type 1 fibers, which are. Cardiac muscle is like a super duper type 1 fiber. They have tons of mitochondria. The mitochondria make up a major percentage of the volume in the cell, actually, in the cardiac cell. And so your heart has great endurance. It may not generate a ton of force, but it does have a lot of endurance. I mean, think about it. That heart has been pumping a couple of times, like 60 times a minute or more, since before you were born. So 20 years, no rest, right? If I do this, or even my hand, if I just do this once every second, I'm probably going to fatigue after a little while. Like, give me like 5 minutes and I'll get either bored or my hand will get too tired. Your heart has been doing that for 20 plus years and it's still going, right? So its energy supply is super sustainable. Another thing, your skeletal muscle, you can voluntarily contract your skeletal muscle, right? I'm telling my brain to do this with the skeletal muscles in my hand. I can't tell my heart to beat faster. I can't say, beat faster, beat harder. I don't have that connection. You can do things that make your heart beat faster or harder, like exercise, but you cannot voluntarily say, contract harder or contract just this part of the muscle and not that part. You don't have that control. Another thing is with satellite cells. You don't have many, if any, satellite cells in your cardiac muscle as an adult. And so damage to the cardiac muscle fibers can be a little more consequential. It's harder to repair, right, without those satellite cells. And so that's when heart attacks and necrosis can be a really bad problem. All right, so how does excitation contraction differ in cardiac muscle? As I mentioned, the action potential is transferred from one cell to another. In skeletal muscle, the action potential is generated on a neuron, which then communicates that to all the muscle cells it touches. And if the neuron doesn't touch the muscle doesn't get activated. In the heart, you generate action potentials here at the SA node. Remember, action potentials are differences in positive and negative concentrations inside and outside of a cell. So you generate this action potential voltage in the SA node, and because of the gap junctions, the flipping of the potential from negative to positive, that's the action potential flipping negative to positive, it keeps happening like dominoes all the way down. If this was skeletal muscle, it would have to stop right there. But in cardiac cells, because of this gap junction, the dominoes keep falling. You keep flipping potentials all the way down. So you generate the action potential here, and it travels through all the muscle that's in the atria, which isn't a ton of muscle, but it travels through it, and then finds its way to the AV node, where the action potentials all converge. And now you have more action potentials traveling down all the way around in your ventricles. So the action potentials are generated and transferred that way. Why would your body be set up like this? The job and the contraction of the heart is very specific. It needs to contract in a very certain way to get the blood out. It has to wring out the heart. The heart kind of does this kind of a motion, just a ringing. And that allows the blood to go out of the aorta. If all of the muscle contracted at once, so if you had contraction up here and contraction down here, all at the same time, it'd be hard to direct blood out, right? You're closing in everything and you might not direct the blood in the right way. So transferring action potentials from one side to another or one muscle to another allows for a very coordinated contraction throughout the whole muscle, the whole cardiac or whole heart. Now the action potentials, how they're generated is slightly different. Remember we talked about how you don't have voluntary control of your heart. You could actually you have nerves going to your heart, right? Your sympathetic nervous system, parasympathetic nervous system can increase or decrease your heart rate. But here's the cool thing about the heart. You could take all nerves off, right? That's what happens with a heart transplant. The nerves are completely removed from the heart. People that get a new heart, they don't get the brain and the nerves of the donor, right? They just get the heart. That heart will keep beating on its own outside of the body or even in the new recipient's body because your heart automatically generates action potentials on its own. So if you remember, in a skeletal muscle, the resting membrane potential was pretty constant. If we don't do anything, it stays under threshold and doesn't activate itself because the potassium and sodium don't leak across the membranes very quickly. In contrast, your heart actually, the membrane's a little leakier, and it lets more positive ions like sodium and calcium into the cell gradually, naturally, without you doing anything. The positive ions flow into the cell, bringing it closer and closer to threshold. And once it hits threshold, you get an action potential, which is passed from one muscle to the other, and the whole heart contracts. Now, if we completely sever the ties with the nervous system, your heart would probably beat somewhere around 100 beats per minute. But we actually have, in a healthy human, we have the sympathetic and parasympathetic nervous system, which slow that down or speed it up. So this slope right here will happen regardless of sympathetic stimulation, so that you would eventually get an action potential right about then, right? Now, the parasympathetic system results in hyperpolarization at the SA node. So you're going to get further away from threshold, which means it's going to take longer to generate an action potential. And every time you get an action potential in the heart, that's a heartbeat, right? So the parasympathetic system makes the SA node less permeable to positive ions. They don't let as many positive ions in, and they'll let some of the negative ions out, or let some positive ions out. Whatever the case is, it just makes the cell more negative and slows things down. On the flip side, sympathetic stimulation, like epinephrine, will bind to beta-1 adrenergic receptors on the heart that bring positive ions like calcium in, and it will get you up to threshold faster. And the faster you get to threshold, the faster your action potentials, the faster your heartbeat. Okay. Now, in terms of once you get the action potential on the heart, in the heart muscle, how do you get calcium release? There's slight differences in the heart versus skeletal muscle. Remember, in the skeletal muscle, we had the action potential that would go and stimulate dihydropyridine receptors. It would go in the T tubules, and there'd be these DHP. Wow, that's terrible. Sorry. It would stimulate DHP receptors, which bound the T tubule to the sarcoplasmic reticulum, along with the ryanodine receptors. And it was the stimulation of the DHP ryanodine receptors that cause calcium to flow out of the SR. That was skeletal muscle. We don't have that in cardiac muscle. We don't have the DHP receptors or ryanodine receptors linking the action potential in the T tubule to the SR. We use a different mechanism. The mechanism that we use is called calcium-induced calcium release. So when you generate an action potential, we still have like the sodium-potassium ATPase pump and all these other things going on just like before. When you generate an action potential and you get a lot of positive ions flowing in, you have these L-type calcium channels. These L-type calcium channels are voltage sensitive. And so when hit up with a positive voltage, they open up and calcium flows in. So you get a little bit of calcium to go into the cell. And that calcium is then going to stimulate the SR. We're not going to talk about the mechanism. Just suffice it to say that calcium flowing into the cell stimulates the SR to release even more calcium. So this is called calcium-induced calcium release. You're using a little bit of calcium to wake up the SR to release a lot of calcium. About 20% of the calcium going into your cell, 20% of the calcium that binds to the actin and myosin, to the troponin, comes from outside of the cell. 80% is going to come from the SR. One of the really cool things we'll talk about in a while when we talk about the contractility of the heart, you can make your heart muscle contract harder by causing more calcium to flow in, either from the extracellular fluid or more calcium to come out of the SR, which causes more cross-bridging. In skeletal muscle, the SR releases enough calcium usually to flood all the troponin sites and get full power, full force, everything cross-bridging. Not the case in the cardiac cell. At low intensity contractions, you just release a little bit of calcium that allows for it to bind to some of the troponin, so maybe you only have half of the troponin bound and half cross-bridging going on. If you add in more calcium, you open up more cross-bridging sites and get stronger, more forceful contractions. So that's something really cool with the heart that we'll talk about later. Now, the cardiac cycle, how quickly you go between heartbeats, all depends on these action potentials, how quickly the action potentials are generated, right? If you have a lot of sympathetic stimulation, let me see if I change this. All right, technology, change it to blue. Say you have a lot of sympathetic stimulation that gets you up to threshold faster. You're going to have heart action potentials and heartbeats much more rapidly. If you go into, whoa, what the heck was that? You guys are totally loving my tech savvy self, aren't you? All right, so if you go into, let's say just a parasympathetic situation, it's going to take longer to get up to an action potential. Right? So every time you see one of these peaks, that's a heartbeat. So sympathetic stimulation increases the heart rate. Parasympathetic slows it down. When you exercise, it's a sympathetic situation, right? Your heart rate goes up. That means you're generating these action potentials more rapidly. Under normal resting conditions, it's usually assumed that the time between heartbeats, that's called your cardiac cycle, that the time between the heartbeats, about 30% or 1/3 is made up of systole, meaning contraction of the heart. And 2/3 of it is usually considered to be diastole or relaxation. So contraction is systole, that makes up 1/3 of the cardiac cycle or 1/3 of the time between heartbeats at rest. Relaxation of the heart, diastole, makes up about 2/3 of it at rest. What happens when you exercise is that now you have, because you're going from heartbeat to heartbeat to heartbeat much faster, you have less, or the systole makes up a greater proportion of the cardiac cycle. So you're contracting for most of the time and only relaxing a little bit. So in exercise, systole makes up a much bigger proportion of your cardiac cycle. And that's going to become important later when we talk about, how did I do that again? I'm just doing this to be entertaining, guys. That's going to be important when we talk about mean arterial pressure. You probably did this at some point in your ex-phys lab or your physiology labs in the past. Mean arterial pressure is often calculated by diastolic blood pressure plus the difference between systolic blood pressure and diastolic blood pressure divided by 3. All right, so if we just have our classic 120/80, this would be, let's see if I can do this without messing up the screen again. Nope, it does it every time. Sorry. Don't know why it's doing this. All right. So if we just say, if we use our standard numbers, this would be 80 plus 120 minus 80/3. Okay, so that's how you can estimate the average arterial blood pressure or mean arterial pressure. Why 3? Well, the 3 is because of the assumption that at rest, diastole makes 2/3 of the time, and systole, which is this part here, only makes 1/3. So that's why you divide it by 3. When we go and talk about exercise, obviously this isn't going to work, because now instead of systole only being 1/3 of the time, it's closer to 50% or 2/3 of the time. So this equation that you've probably seen in the book and in labs, this is good for rest, but not so much during exercise. And we'll talk more about that later. Hey, look, black screen. We're done with this lecture. I hope you guys had a good time, laughing at me at least, and we'll move on to the next lecture. Bye.